Hello, everyone, and welcome to APS Webinars. The title of today's webinar is Miniaturized Biosensors for Healthcare, given by Sufi Zafar, the 2021 recipient of the APS Distinguished Lectureship on the Applications of Physics Award. I'm Crystal Bailey, and I'll be your host for today's broadcast. Thank you all so much for joining us. APS webinars are brought to you as a service of the American Physical Society, connecting you with the expertise of individuals who can offer insight into physics careers, educational programs, and professional development for students, working physicists, and educators. Next slide, please. APS membership gives you easy access to valuable career information and resources, such as this webinar, allows you to get your research out to the community and network with potential employers or colleagues at our meetings, and have a greater positive impact about issues that are important to you through grassroots advocacy. It can also help connect you to a community of like-minded folks through participation in our forums, divisions, and topical groups. If you are not yet an APS member, but value the things that APS provides, we would encourage you to consider joining today, and you can find out more information by going to that URL that's on the screen right there. Next slide, please. So we'll formally introduce our speaker in just a second, but I want to do some housekeeping first. The first portion of our broadcast will be Sufi's presentation. Then we will have a period of Q&A at the end. So if you would like to ask a question, please type it into the questions panel located on the right side of your screen. You'll see the little orange arrow will open and close the panel, and the questions box is towards the bottom. You can submit questions or responses through this questions panel at any time during the webinar. And you can also use this panel to choose whether you connect through computer audio or phone. A link to the recording of today's presentation will be emailed to you after today's event and will also be made available on the webinar homepage. All videos will be available after 48 hours following the broadcast, so please give us 48 hours to get all of that uploaded. We also encourage you to complete the survey upon exiting so that APS webinars can improve its ability to provide you with these valuable services. Next slide, please. I also wanted to mention briefly that APS frequently hosts webinars on exciting topics, such as tips for success in industry careers and career development for international physicists, among many others. So please make sure that you're signed up to get notifications of new broadcasts. By visiting the URL that's on the screen and signing up for the series, you can indicate which series you're interested in, such as the Success in Industry series, career exploration, all kinds of interesting topics. Next slide, please. And lastly, I do want to say a few words about the APS Distinguished Lectureship Award on the Applications of Physics. This award seeks to recognize and honor physicists in industrial and other non-academic careers for their significant contributions to the advancement of physics of a technical, industrial, or entrepreneurial nature, and for their demonstrated ability to give interesting and engaging lectures to both experts and non-experts. Each year, the Distinguished Lectureship Award recipient delivers lectures at APS section meetings, individual department colloquia, and prize sessions at the APS March meeting, and receives a $5,000 stipend. So if you have a colleague that you would like to nominate for this award, please visit the URL that's listed on this slide to learn more. The deadline for the 2022 cycle is June 1st, 2022. Okay, with all of that housekeeping out of the way, let's go ahead and get started. Next slide, please. Sufi, welcome. Sufi Zafar is a physicist with extensive interdisciplinary research experience in biosensors, device and material physics, data modeling, and nanofabrication. She has had a long-standing interest in applying her expertise in physics and engineering to develop innovative biosensors for applications in healthcare, agriculture, and the environment. Towards this goal, she taught herself biology to be an effective collaborator and initiated and led a full-time project on biosensors at IBM Research. Her approach is to exploit CMOS technology tools to achieve sensor innovations with a viable path for commercialization. The Biosensor project achieves significant translational success with tech transfer for commercialization rights to a medical diagnostic company, which she will tell you about in this presentation. SUFI's research efforts have resulted in 79 top-tiered journal publications, 84 filed or pending patents, invited talks, including keynotes, and invitations to serve on technical conference committees. She has received prestigious internal and external awards and honors, including the IBM Research Division Award in 2005, IBM Outstanding Technical Achievement Awards in 2013 and 2018, IBM Master Inventor Award in 2017, IBM Outstanding Innovation Award in 2021. SUFI is also a senior IEEE member since 2014 and American Physical Society Fellow since 2009. And of course, she is the recipient of the APS Distinguished Lectureship on the Applications of Physics Award in 2021. Okay, SUFI, with that, I'm going to hand it over to you. Please take it away.
Thank you, Crystal, for such a detailed introduction. And I also appreciate the opportunity to present a webinar here. Good afternoon, everyone. So today I'm going to talk about miniaturized tiny biosensing devices with application in healthcare. So without wasting any time, why do this? So a motivation, and so this is a brief outline of the presentation. Why do it? So I believe these biosensing devices can have an impact both bettering the society as well as it meets many market needs. I will give you a brief overview of the biosensors, what are the challenges and opportunities. Then I will discuss the biosensor innovations, some examples that we have accomplished here, and then the concluding thoughts. So motivation. Healthcare is very important, and chronic diseases are a global healthcare challenge. Just to get a feel for the numbers, let's look at the numbers. So in US, the direct spending is $3 trillion, which is 90% of the total healthcare budget on chronic diseases. And what are chronic diseases? I have lumped them here in the figure. Chronic, hypertension, stroke, cancer, heart attacks, arthritis. Indirect cost in terms of lost wages is another $1 trillion. And it also impacts the quality of life on productivity. And about 60% of the adults have one or more chronic diseases. And another important thing we have observed that the healthcare cost is going up with time, increasing with time, and that is mainly because of the aging population. And this trend is true everywhere globally, and you can see is percent of GDP and the years, and it's going up. So there is obviously a need for innovative biosensing devices, and so which can be used both as a research tool and also enhance the healthcare and bring the cost down, because very expensive doesn't work. So we need better tools which not only give us a better diagnostics, better monitoring of diseases, but they also lower the cost. And also I would show you these biosensors which we use for healthcare have application in other important, such as sustainable agriculture, environmental monitoring, which are both very important for climate change. So in all, there is an unmet need for biosensing devices. So with that, we set up a goal. Our goal is to develop a platform that can measure different analytes. By analytes, I mean ions, biomolecules like DNA, proteins, either from a biofluid or from a tissue. So we want devices which can be worn on a skin or implanted on a skin for continuous monitoring of various analytes. So this is to understand chronic disease. So for example, glucose monitoring, and also we also want sensors which are so sensitive that they can rapidly diagnose a disease. And if we had such sensors, rapid diagnostics, we could have handled this pandemic a lot better. Say, if you can imagine, if you could just breathe on a sensor, it tells us whether we have got infected or not. So that is another thing. Or early disease diagnosis for cancer and all, so the treatment can start way earlier. So this little cartoon kind of summarizes a goal. We need sensors which can measure the analytes from biofluids such as saliva or sweat. We don't want to draw blood because continuous monitoring is not, so it's better to do it from sweat or saliva. And the sensor device has to be highly automated. It should be able to send the data wirelessly to a smart device like a phone. From there, it will go into cloud. And there we will have other information about the patient, like genetic, lifestyle, environmental, and then using artificial intelligence tool and the sensing data would lead to precision medicine or personalized medicine. So in this stack, if you look, we know how to build circuits. Everyone has a smart device. We know how to send data. Cloud, most of the work has moved to cloud. AI. So what is really missing is sensors which can provide clinical quality data from measuring from sweat or saliva. And if we have all that, then we can be moving. We will understand the diseases better. We will understand how diseases start, and also we'll have a better treatment plans. So that is the motivation. So our goal was to demonstrate electronic biosensors. So of course they have to be high sensitivity, high resolution, and label free. Like optical sensors, they have to be labeled. And because we want rapid and cheap, so we want it label free. They have to be very small because it has to be mobile, carried in a hand or worn on a skin on some part of the body. Very low power because we can't carry big batteries, and highly automated. And even if we get all this, the most important thing to have an impact on the society, it is to be able to mass produce these sensors at a very low cost. Without that, if we demonstrate hundreds of them but cannot volume produce it, they will have no impact. They will just stay within a publication. And our goal was to make a contribution to the society. So to do that, how to achieve this cost-effective mass production and the other goals, we are going to leverage this semiconductor technology, which has brought us, which is 40 years old, plus 40 plus years old, has brought us wonderful products, all our iPhone and laptops and all the electronics you see. And every year you buy an electronic product, it's cheaper than before and better. So how are they achieving? They're reducing the cost. So I come from this industry, so I'm going to leverage the semiconductor in the technology completely so we can have a rapid path to market. So all the sensors I will talk about, they're compatible with the semiconductor technology. That is, they can be volume produced in a semiconductor factory. And so with this goal setting, now I'm going to give you a very brief review of what is an electronic biosensor. So electronic biosensor has three components. It has a reference electrode, which is immersed in your test solution, which could be sweat, saliva, or inside a tissue. And then you have these analyte, which could be a biomolecule which you want to detect. Then you have a sensing surface, which will specifically bind the biomolecule. And then you have the transducer device, which converts the biological or the chemical signal into an electronic signal, and which we measure. So the sensing surface, so here like we see these three components. The transducer device does not depend what you're detecting. It just whatever it senses, it is biological, it converts the signal and often amplifies the signal. Reference electrode is part of your electronic circuit. It also does not depend what you're detecting. The sensing surface, this green layer there where the chemistry, it has to change for every time you detect a different biomolecule or a different ion. So the first two do not depend on the analyte. So there's a lot of work has been done on the sensing surface by the chemists, because this sensing surface is borrowed from the optical sensors. But these two elements, number 1 and 2, there's much less work has been done, huge opportunity to improve them and get better sensors, because once you improve the transducer, the reference electrode, you get a better sensor, because the performance of the sensor depends on all three components. So I will focus in this presentation, because of the time comes only on the transducer device, though we have also done work on the reference electrode. So with that, I will go, what is the sensing principle? So our goal is to be able to detect an ion which is present in a saliva, sweat, or protein, or a virus, or bacteria. And all these analytes have electrical charges on them. And these sensors that I will talk to you about them, they are sensitive to charges. So when they bind to the sensing surface, they change the surface potential. So let's assume this biomolecule has a charge Q. When it binds to the sensing surface, when it binds, it changes the surface potential, which then the transducer converts into an electronic signal. So the sensor is detecting charges, but we want to detect a species of either protein, virus, or something. So that's where the specificity is achieved by functionalizing or modifying the sensing surface after we have fabricated the device. So we fabricate this whole sensor, then as a last step, we coat it with a chemical layer which will specifically bind a target molecule. So you see these biosensors is highly interdisciplinary. It has a combination of physics, chemistry, and biology. And so these are three legs on which it stands. So there are challenges and opportunities both. So now I'm going to talk about what are the transducer innovations we have done for mobile sensing. By mobile, I mean handheld or a wearable or an implantable device. And so at the same time be able to get clinical quality data. So first innovation, so if you want to wear something, it has to be highly automated and the user doesn't have to do much because this is not a trained person working in a lab. Up till now, when we need a blood test, the blood sample goes to a lab and then someone trained does it. But what if you want to do all the analysis out of the labs on the go? So here we have to have a highly automated sensor and also highly accurate. So for that, accuracy depends on something called calibration. You always have to calibrate your sensor. And that we have to be able to do, we should be able to easily do. So right now, people use field effect transistor devices as that's the leading choice. It was proposed 40 years ago. But the drawback it is a complex calibration, which is well suited inside a lab or someone who's trained to do it, but not for a consumer use. So outside, so we wanted to come up with something that will address this and also improve the sensitivity and the resolution of the sensor because we want to detect analyte from saliva or sweat. So we proposed another semiconductor device, which is called a bipolar junction transistor. And I will show you in the next few slides, it has a superior performance as a sensor if you switch just from the BJT to the FET. So what does it look like? Here's the schematics. So this is how the device looks like. It is a semiconductor device. It has a heavily doped emitter, then a base, and then heavily doped collector. And the signal, which is a current, which flows from the emitter to the collector. So these are three silicon which have different dopings. And from the base, you connect your sensing surface. So sensing surface, I'm showing it as a needle, but it can be of any shape. And then you have this reference electrode, which completes the electrical circuit. So for different analytes, you'll have to change the coating on the sensing surface. And here's the equation of the signal. So when the surface potential delta psi changes because an analyte has bound to it by delta psi, it exponentially changes the signal. So it's not only detects, but it amplifies the signal. So that's why it's superior. So we are going to, because FET are extensively used, their products, and we were proposing something new. So we do a very detailed comparison why BJT transducer, these new transducers are better transducers. So here, so this is an FET. So we took two sensors which have the same sensing surface, and in this, which is this red needle, we call it a silver chloride, AgCl surface, which are sensitive to chloride ions for comparison purpose. And then we took a FET device and a BJT device. Both were fabricated in IBM. They had the same area. They were fabricated state of the art. So, and we are going to sense chloride using a silver chloride surface. And then we use the same reference electrode. So they are identical, these two sensors, except the transducer device. In one case is the FET and the BJT. And then we do identical measurements. So first we look at the calibration, which is critical for getting clinical quality data. So first, I'm going to show you signal as a function of applied voltage. So IC is the signal, the collector current, this y-axis is the signal for both the cases. So here is the data for the BJT sensors, and we did the measurement in different concentration of chloride. And the chloride, we observe as the chloride concentration, the curves are shifting towards the right. For the FET also, we see the same thing happens, but you notice the shape of the curves are different. Here, for BJT, the current rises exponentially, whereas here they saturate. And this difference has a significant impact both on sensitivity and calibration and resolution, and as I will show you in the next. And so I looked at the calibration curve. So I've normalized my sensing current for comparison purpose with 1 millimole of chloride concentration. So the solid curves are the calibration curve for the BJT measured at different applied voltages. So I'm basically taking the data at fixed applied voltage. And you see they're on top of each other. And the slope is steeper. Steeper slope means it is more sensitive. For FET, the shape of the calibration curve depends very much on the use voltage. So an user has to make sure that they are using at the right use voltage to get the optimum sensitivity. And during use, if the signal has drifted and it has come to this calibration curve, then you'll start to get errors. Whereas here, if the signal has moved, you will still have the same calibration. So BJT is much better for continuous monitoring, as you need for chronic diseases to understand the efficacy of drugs when you take, or so for something a wearable. So that is why BJT has this one advantage. It has a more robust and simpler calibration, which does not depend on the use voltage. But both of them have a use voltage less than 1 volt. So they both are low voltage and low power. So next, the other thing is sensitivity. So sensitivity is defined as the relative change in the signal per unit change in the voltage. So FET has a shape. You see the FET sensors rises up and then comes down. BJT, as a function of the sensing current or sensing signal, is flat across 6 decades. And it doesn't depend. So which is, again, it doesn't matter. Initially, whatever your sensing current was, your sensitivity will remain same. And during the use, the example, the signal has moved to another point, your sensitivity remains same. Whereas here, while your signal, due to some changes, has moved, the signal has increased to this, your sensitivity decreased, and now you need to recalibrate and correct it for this signal. So every signal you need to calibrate. So that is a problem if you want to take the monitoring of a disease outside the lab in the form of a wearable, and this BJT tends to solve it. The signal to noise ratio is another important thing. So here again, signal to noise ratio does not depend on the sensing signal, and so your signal could change during the measurement process because your body is changing or whatever analytes or something, it will remains the same. Whereas for FET, it has a peak. And you notice one thing, the peak of the sensitivity and the signal to noise ratio are not at the same point. It peaks at 10⁻⁷ amps, whereas here it peaks around 10⁻⁻10 amps. So you always have to do a trade-off between sensitivity and signal to noise ratio, which means resolution. So just signal to noise ratio basically translates into resolution, which means how accurately can you measure. What is so here to show you resolution, just not to talk in abstract terms of signal to noise, we did an experiment. We took 10 millimolar of potassium chloride and diluted it in steps. And as we diluted it, we saw the signal increase. And we repeated both the experiments for both FET sensors and BJT. Here we see as we dilute, we can clearly resolve this small dilution in very clearly. So you can see the step is 5 milli of delta peak chloride. So whereas for FET, we cannot resolve it at all. So it is at least 8 times more. You can see the noise is so much smaller, and so we can resolve our smallest changes. So that's another important thing. No trade-off, robust. So we believe BJT sensor is an improvement over the FET, and We have published this, we have patented it, and they have companies who are now interested in monetize into bringing into products. So another important thing when it comes path to the market. So one thing is to publish the data where you can have 5, 6 sensors and show your results. But to make a product is harder. And one of the thing is sensor to sensor variation has to be minimal, because if you are shipping out 1 million, 10 million parts, you cannot sit and calibrate them, check them, all of them. So you want to make sure all the sensors that are getting manufactured, they look identical. And here again, BJT has an advantage. So BJT is inherently has small variation in sensitivity, which is an important parameter. So here I'm showing you sensitivity for different BJTs. So the open symbols are the BJTs manufactured in IBM. Solid line is a BJT I bought from commercially available. I bought it for a few penny from the market. And you can see they both have, their all sensitivities look very similar over several decades. Whereas for FET, it depends who made it, how they made it, and then the curves, the sensitivity curves depends very much on all those details. So inherently BJT has a less variation, which is another strong point for commercialization. So now I'm going to show you some applications, because this was to show that it is a better transducer, and it doesn't matter which analyte you want to detect. If you have a BJT, it will give you a better performance for a wearable, for example, or implantable, or where you don't, something where you want to do continuous monitoring. So we started to look, because this was a chloride sensing, so we looked at artificial sweat and to measure the chloride. The chloride so cystic fibrosis is a chronic disease, and they diagnose this disease or monitor it by measuring chloride from the sweat. So that's why we did this. So first we established a calibration curve, which is the open signals using potassium chloride signals. And then these are the measurement of chloride in different samples of sweat with having different chloride concentration. And you can see it is reversible. And from this calibration curve, we read off how much chloride is present in each sample. So, and then we did the comparison, the values that were provided by the vendor who made the chloride artificial sweat samples with different, and what we measured. And you can see this is sweat here. They have good, is a very good, very well matched with the vendor provided. Another important thing which impacts the sensitivity is the stability of the signal. Often the signal can start to drift in a solution for many reasons. So here we're showing for 10 minutes we could measure. You can see the noise is 1 in 1,000 part in a sweat, because sweat has other proteins and other things which can stick to the surface and cause problems. And we see very low noise and very stability. So showing, and also we use very low voltage for is less than 1 volt. So this shows one application which we reported. So that these measurements were done on IBM's fabricated, but we wanted to see, can we just buy for a few pennies in from the semiconductor component a BJT device which is available for a few pennies for a company like DigiKey or any electronic online store, or any store, electronic store, online or otherwise. So we bought one, and you can get very small ones, but I had to handle by hand. So I bought this one, and I use this to measure pH. pH is pH is proton concentration in a solution, very important for healthcare. So I tied the base with a needle which was coated with titanium nitride. It is a CMOS compatible material, that meaning it can go into a semiconductor factory. As well, it is a biocompatible material, so it will not cause biofouling. And then we started to look at the pH sensitivity. So it looks exactly what we had observed for chloride, because we have changed the transducer, and transducer doesn't depend on what you're detecting. So here again, I'm comparing the BJT sensitivity for pH versus FET, which is the data. Again, you see sensitivity is better for pH sensor with the BJT transducer. Signal to noise ratio is also better, and they both peak for the FET like we had seen previously, no trade-off, same behavior. So we have a higher sensitivity PA sensor and signal to noise ratio. So we wanted to apply it. So here I'll show you an example as a research tool. So I had a professor visiting from Columbia, and he was trying to improve the enzyme called glucose oxidase activity. So glucose oxidase, when glucose interacts with this enzyme called glucose, it produces 2 protons and 2 electrons. And this enzyme is used in all glucometers. Presently, the glucometers, they count the amount of electrons that are coming because there are no sensitive PA sensor. But since we had it, I decided to count the protons. And he wanted to know because if you have an enzyme which has a high activity, you improve the detection, you make a more sensitive glucometer. So what he was doing, he was making DNA cages and attaching the enzyme outside the cage, inside the cage, and he had a control. What he found in Columbia doing experiment, optical, that's what they had the best, that there is an improvement. The yellow bars are the optical measurement, measuring the enzyme activity, and they see there's some small improvement, but they could not distinguish between internal attachment and external attachment. But when we repeated the same experiment using this few pennies biosensor, which cost a few pennies, we could clearly show that when you attach the enzyme outside your DNA origami, you have a higher activity. How did we measure the activity? What we are measuring, so we did an experiment. We had this enzyme attached to this. So we did an experiment where we are monitoring the proton. At t equal to 0, glucose is added. When the glucose is added, the enzyme starts to produce protons and electrons. We monitor the amount of protons. So this is for the control. This is for the internal attachment, external. And then if you differentiate these curves, you get the activity. Activity is defined as the change here. This is given by this. So we have published this data in ACS Nano. And so with this, so this shows as a research tool to measure small things. And also it can go for glucose sensing. I'm going to give you a third example. And this is the detection of DNA. So this is, again, I did not use a best state-of-the-art BJT. I just took the same BJT which I had bought for a few pennies. Now I attached a gold tip to it. And I wanted to see that if I could measure in real time hybridization of a DNA. So it is, again, an example as a research tool. And so for biomedical research tool. So we took a sequence of oligonucleotides, and it has 25 nucleotides. The sequence is important for, I believe, lung cancer that I was told. So first, and it has a thiol at one end, 5' end. Thiol binds to gold, so that's why we are using gold. So we put the gold tip, we dip it in a solution which has this oligonucleotides dissolved in it, and as it starts to self-assemble on the gold surface, and remember DNA is highly negatively charged, the signal starts to decrease. As it's become more and more, it decreases, and we could measure it, and we could say about after 2 hours it has saturated. That means self-assembly has occurred and it has completed. All the DNA has self-assembled on the gold surface, the single strand of self-assembly. So now we wanted to verify. So we used XPS. So we took the sample out, measured a sample which had no self-assembly versus something that had undergone self-assembly as in this experiment. So the gold one is bare gold, and we pick up a phosphorus peak. So DNA has both phosphorus and nitrogen. So we also pick up a nitrogen peak. And in a DNA, theoretically from the sequence, you can calculate the ratio between nitrogen atoms and fat is 3.6. And if we divide the peak heights, we get 3.5. So it not only we observe the phosphorus and they are quantitatively in agreement with what we expected from the theoretical value. So that's showing that we are measuring the self-assembly. Now we wanted to hybridize this self-assembled DNA. That means we wanted to combine it. So we took another solution where it had the complementary DNA strand dissolved in the solution. And we started to, we saw the signal coming down. We said, okay, it is hybridizing, signal is coming down because this signal is also getting more negative charges. Signal decreases, implying more negatively charged DNAs. But to our surprise, the signal started to go up, and we repeated the experiment and still start to go up. So why did it go up? One is everything is dissolving, coming out into the solution. We did XPS. We saw more nitrogen and phosphorus peak. That means more DNA was accumulated. Why did the signal come up? So one idea was that the DNA is changing its conformation. And so let me once say, all these electronic sensors can detect everything which is within a debye length. What is a debye length? It's this perpendicular is a distance within which it can detect the charges, and beyond it, all the charges are screened. And debye length depends on the ionic concentration of the solution. Lower the ionic concentration, larger is the debye length. So we changed the ionic concentration and started to measure the signal. We found when we looked at the single strand DNA, the first experiment there is no dependence on the debye length, which means the negatively charged DNA is lying flat on the gold surface, which is understandable because of the electrostatics. But when it is a hybridized, a duplex DNA, it depends on the debye length. That tells us that the DNA after hybridization is not lying flat, but is standing up. And it is standing up, so we see the signal saturates at about 25x. If it was perpendicularly standing up, then the length should have been 78 angstrom. That means it's either highly inclined or bent down. So I collaborated with one of my colleagues in IBM, Payal Das. She did molecular simulation, and these results were confirmed by that, and it showed that it does. So this is another example, and we also built prototypes to show that you can use and do that. So this was more to show the commercialization part of it. We built a prototype, and these sensors, these BJT sensors, this is the whole automation circuit, all built with off-the-shelf. The total thing costed like $100. You can bring the cost even lower, and you can also build it on a single silicon chip in a semiconductor fabrication if you want to produce millions of it. And you can then hook up this whole chip either to a smartphone, or you can do this is with through a cable, because we're using the battery of the phone. Here we use our own battery, so it is a wireless, and you can get high quality data. So that was it. So I end this, and I go to the one that has already gone into commercialization step. As we speak, it is getting manufactured in a foundry. So I have told you so far how great these BJT sensors are, but they cannot do everything. For early diagnostics, you need FET sensors. Early diagnosis, and the reason is I will explain you very quickly in a slide. So the BJT sensors are great for continuous monitoring and other things. But if you want to do single molecule detection or something in attomolar, then you have to go back to FET type of sensor, a FinFET, and I'll talk more about it. The reason is to detect at low cost, you have to miniaturize your sensor to the scale at which a protein is. So that, and BJTs cannot be miniaturized below 100 nanometers in a manufacturable, because I only want to create sensors which can be mass produced at low cost, because our goal is not only to do good science, but also to positively impact the society. And also, of course, I work for IBM, good for IBM. So it's for both society as well as for my company. So I will also, but FET in fact, the cell phone you carry right now has a device which is about 10 nanometer or 14 nanometer or smaller. So they're already manufacturing that size length scale. So I wanted to do that, and we have licensed this whole technology to a company called NanoDX, and it is getting mass produced in a semiconductor foundry called Skywater. So why nanowire? Why nanowire? Why nanos? Because FET can be miniaturized well below 50 nanometer. And that is important when you want to do early diagnostic, or you want to have high spatial resolution. Spatial resolution is just for more as a research tool. And then so why do for ultra low analyte concentration, signal to noise ratio equation becomes this. It depends on the under root of the area. So you need to have a sensor that's area is very small, which means you have to miniaturize it. And that is why we had to go back to the FET for early diagnostics, because we cannot mass produce. We do not have a process for BJTs manufactured. So nanowire have been worked for over 20 years, but they have not come as a product yet. They have numerous papers which show how transformative they can be for diagnostics. And one of the reasons that was holding them, there was a large center to center variation, and that was limiting. So when we got in, I decided to work on this problem. And this is a non-trivial problem. When you make your sensor 30 nanometer, making them all alike becomes very hard. It becomes really hard. So that is why, that's why you have to use a lot of science to solve that problem. And I will talk to you about how to fabricate these sensors. So first, it's not only fabrication, the design has to be such that it makes it. So people were making these round wires. Round wire is not a manufacturable process, meaning you cannot give it to a foundry or a factory and say make wires. But what you can make is a fin, the rectangular shape, a bar. That is a manufacturable. So it's a fin. So we don't use nanowire. We lose there's some surface area loss, but the gains are a lot bigger in terms of sensor to sensor variability, as well as manufacturable process, that which is very important, cheap manufacturable process. So we made the length to be about 1 micron long, and I believe our hair thickness is about 50 micron, if I remember correctly. So that is and 1 micron is 1,000 microns make a 1 nanometer, just to give some of you the ideas of the length scale. And width and height is 30 nanometer. So a protein is like 50 nanometer. So we want to, the size could be we used a surface, the sensing surface is a dual layer of dielectric, which is silicon dioxide, which is gate, and hafnium oxide. This is a new material which was introduced, and I'm sure all of your iPhone has this new material because it allows us to bring better higher performance chips with this new material. So this reduces the defects, which gives us a better sensitivity and resolution. This allows us to reduce the use voltage and also prevents sodium diffusion inside the device into the transducer during the test time. So we did experiments, and our simulation showed us that the best sensor to sensor variability is minimum beyond 30 nanometers. So we didn't go to 10 nanometer. We fixed it at 30 nanometer. So with that, we wanted to, so another important thing we wanted to detect at very low, to remember another important thing for ultra low concentration, there's the sensor area, this fin area is 10,000 or even more smaller than this passive area, the area between the sensors. So if you have very low concentration, and if all the surfaces of the same type, both the sensing surface and the is of the same material, then what will happen if you put a very low concentration analyte? It will bind between the sensors because this area is much bigger. So the probability of binding is much higher than the sensing, and no detection will happen. That implies that when we build a sensor, we must make sure, as I show in this picture, the sensing surface material and the passive surface material should be of two dissimilar materials. So that we chose hafnium oxide as a sensing surface, and passive surface also we made it SiO2 silicon. And these are the surfaces which will be in contact with the test solution. Then we had to develop the right chemistry. So the silicon fin is covered with hafnium oxide, and that is a sensing surface in contact with. We synthesized a molecule, we read the literature, and we found a molecule called hydroxamic acid, which likes to selectively bind to hafnium oxide, but not to so this is the special coating you need to put before you can detect the protein. So this Y is that special coating you need to do after you have fabricated it. And to this Y molecule, we add a ligand, another biotin, which will selectively bind to our test protein, which I'll show you results later, streptavidin. So this is a scheme. So you have to dissimilar material, then develop the right chemistry so that only this Y molecules only coat hafnium oxide, not between, because that's what is needed for ultra low diagnostics. So now the fabrication challenges. So these are TM and SCM images. So when we started out, so we want low defects. So, and if you have a very rough silicon fin, you're going to get a lot of defects. So we optimized a photoresist. I will not spend too much time because it will take, either because of the time limitation, but we optimized the photoresist, and that gave us nice, smooth fin walls. With, and we also removed the damaged, so these fin walls are made out of fin walls. We removed the damaged by etching it out, so we got a defect-free smell wall. We looked under TM, and you can see before and after. It has a nice smooth curves, nothing here. We also found regrowth happening when we were implanting, so SiO2 layer, which is in contact with the silicon fin, but not the sensing surface, but is not the sensing surface. It is not uniform in thickness. When we saw during the it was regrowing, and we found the problem that oxygen was diffusing in. We fixed that problem. Then we found we were using silicon nitride as a hard mask. We found it was not getting completely removed. When we tried to over etch, we was removing the hafnium oxide. So we found it was a problem with our optical alignment, and we fixed that problem. But after all those problems, and here these problems were already solved by the CMOS industry for building these chips, which we all use. So I didn't have to reinvent the wheel. There is a vast knowledge already there. All that I had to do, tap it and rapidly use it and keep the cost down, and yet come up with a superior sensor. So don't reinvent the wheel, but so we are reusing the technology that was used and adapting it for our use. So here is a TM cell image. Beautifully, you can see the silicon atoms here, and you can see a nice smooth surface. Here is the hafnium oxide, and this looks beautiful. And so we have published all that result, but you can see we are looking at the atoms and how smooth this is. So, well, this is all metrology. The real proof of the pudding is in electrical mesh. So that's what we go up to. So I'm now showing you how do we do electrical. So this is we have a chip where we have 24 individual these FinFET sensors. Each is 30 nanometer wide and 30 nanometer high and 1 micron long. Okay, and so here you see an 30 nanometer, and this is size of a single protein, or even a lot of proteins can be 10 nanometer. So we are right on the size of a single protein, the width and the height is. And you can see that each wire is, so there are 25 in, so we have packed in a small area, a lot of them. So we can do test 25 of them all at the same time, take a mean average, so you're not, so you can get better errors if there's a false positive that can be or detected through that. So now you put the solution on top, you have a reference electrode, and you do electrical measure outside the solution. You don't want there. So this solution is only confined over where the sensors are. Everything else is coated with glass, SiO2, and these are where the electrical contacts come. And we do all for benchmarking, for comparison, we are doing in pH buffer. So first we wanted to see how a process has improved everything. So this is the signal, sensing signal as a function of applied voltage to the solution through the reference electrode. And then here you see the reference to sensor were huge, 380 millivolt all over. After improvement, it has gone to 88 millivolt. And we see also huge improvement into on, we call the lowest signal, off signal, on signal, huge improvement. So using this data, now we look at the FET characteristics and the sensors. It's something called how steep is the slope. This slope determines both the sensitivity of a sensor as well as the resolution. So we want the slope to be steep, and we call that subthreshold swing. So this subthreshold swing, which is the how much is smaller, the better. So after improvement, we get the theoretical limit, which is 60 millivolt per decade of change in the signal. Open signal is before we optimize the process. You see, they are all over. It's about 100 millivolt. So and this is theoretical. And solid symbols are from the literature, and they also get the same what everyone else was before we optimize both design and fabrication. VT is defined as the voltage at which the current is 1 nanoamp. So we read off. Here again, we see VT has become much more tighter compared to what before. There's another important property which depends on transconductance, which you obtain by differentiating these curve, which is DID, DV, so we see again very little sensor to sensor variation before we see a lot of variation. So, and then we see hysteresis. If you do a sweep up, sweep down, they come on top of each other. Here you see they're shifting. That means some charges are building up, and they all will. So these measurements were done each 70 devices, and then 70 before and after, so total 140. So now we looked at the sensor sensitivity, very important. We see this is after optimization. The shape of the curve will remain same because the device physics remain, but you see it rises and these 70 devices on top of each other. Whereas before they were all over, they were changing. And here is the data from the literature, and you can see they all have different labs, different data. We look at the calibration curve, they are on top of each other after we did, which is very important, whereas for every sensor has a different calibration curve before. We look at the signal to noise ratio, it has significantly. And all this property, if you think about the device physics, is about the defects that are at the interface of silicon fin and the SiO2 gate dielectric. And we want to minimize. We see after we had minimized the defect, which improved our the transistor properties, both sensitivity, calibration, all these properties are basically physically tied to the defects on the interface and before. And we see, and also I want to point out, smaller is the width of your sensor, higher because your walls of the fin form the greater areas to the defects. So what we have reported, the narrowest sensor with the best defect compared to what is in the literature, is above 80. So what we had, and most importantly, this whole FinFET sensor can be fabricated in a semiconductor plant, and that's what attracted this company to us. They came to us, we have licensed it out, and they're manufacturing. So it is now, they are manufacturing as we speak. They are, and they will get the FDA. And I want to, if this is not, so they can detect, they plan to detect, as their website talks about, they want to detect traumatic brain injury markers, head injuries. They want to detect using this in different viruses for future pandemic, because once you manufacture them, then you coat them with different chemical layers and detect different things. So you have a platform where you can do that. So that's what they plan to do. And they, before they licensed the technology, they took, I gave them the chip and they detected COVID on it, and they were very happy. And that's how, so that's what they did. So now I will do, but I was looking after, I wanted to detect a single protein, and my calculation told me I should be able to look at a single protein, study the physics of it, how it stays, what is it doing on the surface when it. So to detect protein, you have to coat the surface for surface functionalization, which is a big. So to do that, you have to clean the surface. Nothing will self-assemble if the surface is dirty. So we did a lot of study how to clean the surface. But again, we were looking at processes which will not damage the sensor, but will give. So we looked at different, and all this learning also has been transferred to this company, and they're using our processes. We also found it takes 8 minutes to dirty the surface. So we now do a self-assembly in a clean room environment to get good results. So once you self-assemble the hydroxamic acid with that ligand I showed you, then you are ready to detect. You can do on many, many batches, and then can store them in a dry air, in a dry box, and just use them. So we started to look at protein detection. It worked very well for higher. 10 femto. Femto is 10 minus 15. I wanted to go even 1,000 times lower, but I could only detect 10 femtomolar, and above that, I could do above that, but nothing less. And that was very puzzling. So we were detecting this protein called neutravedin, and we could not. And the reason is, when you have such low concentration, you have to wait longer for that protein from solution to find its way and go and bind on these sensors. So your incubation time increases. So it increased to half an hour. And what we found during that incubation time of half an hour, the whole film started to dissolve and we were unable to detect. So we started to work and we verified it that it's dissolving by using XPS. So before incubation, this has a nitrogen peak. We were using that. We saw before putting it in incubating in solution, we saw nice big nitrogen peak. Half an hour later, it was all gone. So, and we have a pattern how to fix it, and we are still working on single protein detection because we want to use it for other, and also to understand science as a science tool. So this brings me, I think I'm doing okay. This is what the goal is. Have electronic biosensing devices which can be cheaply volume produced. That is the key for impact, because it's not just to publish. And then we believe healthcare, as I showed you, but I'm using these very sensors for agriculture because, you know, food security is very important. As climate change becomes, we're going to need, people are cutting down more forests, which is making climate change worse. And if we want to improve food security, better agriculture, we need to go to precision agriculture, use less, get more yield out of it. And that's where we need to be able to measure how to put just the right amount of fertilizers, nutrients in this. So we can use these sensors, especially the BJT, we are applying them for agriculture, for environmental monitoring. So I believe these, and this whole work, it reuses and leverages this vast expertise that was developed over 40 years in the semiconductor. And I believe the convergence of silicon technology or the semiconductor can really transform many of these areas. At semiconductor tech has transformed how we study, how we communicate and entertain ourselves by all these electronics that we use every day. And finally, I want to acknowledge my collaborators, Chris Demick. He was the integrator. He fabricated the wafer, this fin sensor. Ali was the chemist who I collaborated. Ashish was my postdoc. He has his own startup now. Julia had worked a master's program with me. Pyle was a collaborator, and Takning he's retired, but he was an IBM fellow, and I learned a lot from him. Then, of course, the labs. And then this is a picture of a beautiful lab. And it's a very beautiful building, besides having lots of smart people. And thank you for your attention.
Great, Sufi. Thank you so much. That was really, really interesting presentation. We have several questions that have come in. I think a few of them have been answered, most likely. Some of them about, you know, particulars of the probes and the materials and things like that. So I think we might unfortunately only have time for maybe one or two questions. I guess I'll start with one that came earlier that was asking if you have sensors for for ribonucleic acid. I mean, I think that probably refers to DNA, but they did say it.
Yes. So I mean that whoever, if they want to contact me on my IBM is there, they can send me an email. And yes, so these sensors are not just for one analyzer. So that's why I was trying to show we've improved the transducer, improved the overall sensor, and then by changing the chemistry, we plan to detect different things at much higher sensitivity and resolution.
There's another question here. It's kind of more of a technical question. Can you trade off longer incubation time by using a larger detector area to improve sensitivity?
No, so the sensitivity depends on the area. That's why I went from BJT to FET. You have to miniaturize the sensing surface. And I wrote that equation. So there's a signal to noise ratio. So you can't, but you can have arrays of them. So you do arrays of them. You can have hundreds of them, and each of them are very small area. And then you do parallel sensing, and you could do that. Yes. So but you are now using multiple, you can have a vast, like 100 sensors all over it. So that is one thing. And then there are some other ideas, use electrostatic to pull your target molecule towards the surface. So there are some other ideas too.
Great, very cool. So maybe one last question,
yes.
Rather than getting into the technical things, I think someone was curious. So it says in the end, you know, you mentioned that you published results in some of the journals,
Yes.
but the assumption in industry is often that you can't really publish out of concerns about IP and ownership of IP. Can you say a little
No,
bit about how
yes.
the process works?
IBM publishes a lot. I mean, IBM has 3 Nobel Prizes and Turing Awards and all those things. So we do publish, and and I came to IBM because of publications. I was so impressed with the publications in IBM. So that's a hiring tool also. So we do, IP is very important. So what we do first, we apply we make disclosures and apply the patents. And once you have filed the patent, then we go and publish. So I think people think you can't do what, you can do both, and it all works. So all this first we applied, and once you've applied, you have filed the patents, because the patents become public after a year, so you can publish. So once you file the patent, you can publish. So that's what we do here.
Got it. That's very good, concise answer to that question. Okay, so I guess one more question, and then we will need to wrap up.
Yes,
So if anyone in the audience today was interested in doing something similar in their career pathway,
yes.
working at IBM, I mean, what would be some advice that you would give to someone who wants to kind of follow in your footsteps and work on similar things?
So, okay, the short, I have signed up for Impact, Crystal, so they can just contact me, and I'm at least supposed to spend 1 hour mentoring, so I can do that. Because without knowing what the person's background is, I mean, I can, so I'll be happy to chat with that person. They can contact through Impact. I have signed up for Impact. So, and otherwise, I'll say a physics background really helps, because you can see how interdisciplinary, and I believe having physics background helped me a lot.
Again, a wonderful, concise answer, Sufi.
Which you like.
Yes, of course. Well, especially we're out of time, but I do want to provide a little bit more detail. So what Sufi is referring to is the Industry Mentoring for Physicists program. I am putting the URL in the chat. So Sufi and many other wonderful folks from industry have volunteered to mentor students who want to log in. You do have to be an APS member to access it. But once you are in as an APS member, you can go into the database, you can search on different people in industry working in all kinds of different areas and get in touch with them. These are folks who have signed up, who are ready and willing. They stand at the ready to answer any questions you have about working in industry, help you pursue those career pathways. So with that, I'm going to go ahead and wrap things up. So we apologize if we didn't get to your questions. You can certainly follow up by sending an email to webinars@aps.org, and we'll forward them along for comment or for try to get your questions answered. I also want to remind you, everyone who's here, that a recording of this video presentation will be sent to you via email and will be made available on the webinar homepage. So aps.org/webinars. It's all on YouTube. So we do ask you to allow us 48 hours for us to complete that upload. And lastly, before you sign off today, it should launch a survey. We really appreciate it if you take a few moments to give us feedback. So with that, I'm going to thank Sufi again for the wonderful presentation. And thank you all for coming. And we'll see you next time. Have a great day.
Thank you.
All right. American Physical Society, copyright 2022, all rights reserved. Bye.
Bye.